Hello everyone, I'm Rafael Terra, and I'm going to present a work entitled HP2Net Empowering Efficient Phylogenetic Network Analysis Through High-Performance Computing. Starting with the problem statement, high-performance computing has enabled increasingly complex genomic analysis, including those in phylogenomics. In phylogenomics, the evolutionary relationships between a set of individuals are typically represented as phylogenetic trees. However, trees are not always the best structure for this purpose, as they cannot represent reticulate events such as hybridization, recombination, and horizontal gene transfer. In this case, phylogenetic networks are more appropriate. Constructing these networks requires significant computing power and several steps, since the software used for network construction often relies on inputting data from previous phylogenetic analysis. Therefore, performing this analysis manually and one by one can be error-prone. To overcome this challenge, we present the key contributions of our work. First, we introduce HP2Net, a framework for phylogenetic network construction built on state-of-the-art software. HP2Net can execute multiple workflows and datasets in a single run. The framework is designed to run on both HPC clusters and local machines and includes mechanisms such as data reuse and task packaging to improve performance. We also present an exploratory performance and scalability analysis of the software used by HP2Net, identifying bottlenecks and evaluating the behavior of multi-threaded applications. Finally, we showcase a case study based on the dengue virus genomic data, demonstrating HP2Net's applicability in a real-world scenario. HP2Net was developed using Python and the Parcel library to orchestrate the workflows, and Parcel was chosen, among other reasons, for its portability and ease of use. The framework takes as input a set of multi-sequence alignments of genes from the same individuals and offers 5 workflows with distinct methodologies, using XML, IQ3, and Mr. Bayes for tree construction, and PhyloNet and PhyloNetworks for network construction. Here you can see some results from the scalability analysis of the framework. This analysis showed that executing HP2Net with all the 5 workflows is comparable to executing only the slowest one, and the impact caused by the data reuse when comparing HP2Net to the sequential execution of the 5 workflows. In our case study, we analyzed the dengue-1 genomes obtained from GenBank. Focusing on Brazilian samples, we selected 43 complete genomes for genotyping, phylogenetic reconstruction, and network analysis. This dataset was reduced to the 10 most representative genomes, each annotated with 10 genes. These were aligned and used as input for HP2Net to construct phylogenetic networks using the 5 workflows, demonstrating the framework's applicability. In conclusion, we presented HP2Net, an open-source framework for constructing phylogenetic networks in HPC environments, accessible via the QR code below. HP2Net proved to be a robust and flexible tool supporting various methodologies. It showed a reduction of 90.96% in execution time compared to a sequential execution, and data reuse contributed to a 15.35% reduction in runtime. Our analysis of dengue-1 genomes revealed potential reticulate events. However, further research is needed to confirm these findings and fully understand the implication.